Okay, we can start now, I think. Distinguished guests and esteemed colleagues, I'm delighted to welcome you all to the 8th session of Cerrahpaşa Neuroscience Talks. Before starting the seminar, I'd like to say a few words about today's importance. Professor, is that all right with you? Okay, today is the March 14th, Medicine Day. It's the opening date of Tıphane Amire, the first school of medicine. In 1827, celebrating Medicine Day every year represents the official transition of our medical education from Eastern medicine to the Western medicine. Today also holds an important place in our history too. During the British invasion in 1919, a med student named Hikmet Boran and his friends wanted to celebrate this date by hanging a Turkish flag between two towers of the faculty building. The occupation forces were terrified with this view. Even though they tried, they couldn't take down the Turkish flag. So since that day, we have been celebrating March 14th as Medicine Day. So firstly, I want to say Happy Medicine Day to everyone here. Okay, today we have the great privilege of hosting an exceptional scholar and influential expert in the field of neuroscience and neuroengineering, Professor Miguel Nicolelis. Our focus today will be on intriguing topic that lies at the core of our understanding of brain-computer interface. Brain-machine interfaces, from basic science to neuroprostheses and neurological recovery. As we eagerly await the insightful talk by Professor Miguel Nicolelis, let us first acquaint ourselves with his illustrious background and accomplishments in the realm of neuroscience. Professor Miguel Nicolelis studied medicine and obtained his PhD in neurophysiology at University of São Paulo School of Medicine. He is now a distinguished Professor Emeritus of Neurobiology at Duke University and founder of Duke Center Neuroengineering and the Edmund and Lily Safra International Institute for Neuroscience of Natal. He's also the president of Nicholelis Institute of Advanced Brain Studies. Professor Nicholelis has dedicated his career to investigate how brains of freely behaving animals encode sensory and motor information. His research in biomedical engineering, robotics, and neurophysiology has influenced many neurologists around the world. And he's been a member of Pontifical, Brazilian, and French Academy of Science, and he has authored over 160 manuscripts and holds 3 US patents. It is an immense privilege to have Professor Dr. Miguel Nicolelis as our esteemed guest, and we eagerly anticipate the wealth of knowledge and expertise he will share with us today. After his presentation, we will have a Q&A session where you can type in your questions in the chat box. So without further ado, I welcome Professor Miguel Nicolelis, and I'm looking forward to him to enlighten us on captivating topic, brain-machine interfaces, from basic science to neuroprostheses and neurological recovery. Professor, welcome, and you had word.
Well, thank you very much for your kind introduction and for this wonderful invitation. Happy Medicine Day to everyone. It's a great pleasure to join you from Brazil today, from São Paulo. And I hope I can give you in the next 50 minutes or so overview of this field that was created 25 years ago in my lab, and that now is all over the world and offers a lot of hope to millions of people, hundreds of millions of people around the world that suffer from untreatable disorders of the brain. So without too much, I'm going to share my screen here. Please let me know if you can see my PowerPoint, and we can go for it.
Yeah, we can see it right now, Professor.
Oh, you can see it. Wonderful.
Yeah.
Wonderful. Yes. Basically, as you heard, wonderful. As you heard, everything started in this field when we acquired in the mid-90s the ability to basically record. Oops, something is happening here. Uh-oh. I got it. When we acquired the ability to record large numbers of neurons, as you see in the left part of this screen, simultaneously from the brain of animals. This was the first major recording of about 100 neurons, the electrical activity, extracellular electrical activity of about 100 neurons is spread over 5 different cortical regions of a rhesus monkey that was awake and doing, arm movements while we recorded her brain. And you can see here in each line, the activity of individual neurons. These vertical bars are action potentials being produced by each neuron over a period of about 10 seconds. And it was this possibility that opened up a lot of different spin-offs that I can show you in okay. Something is happening here that never happened before. My computer is not accepting the commands here. I don't know if it's because, let me see. Can you still see the screen?
No, it's off.
Oh, it's not there.
No.
Huh. This is strange. The first time I ever had this kind of problem. Let's see. Okay. We'll see how it goes. This is just a summary of everything that came after we developed this method for chronic multi-site many electro recordings. That's the way we name it. The technology that allow us to implant in multiple areas of the brain these very fine filaments that we call microelectrodes that basically allowed us to monitor large numbers of neurons, hundreds of neurons simultaneously, while animals did all sorts of behavior tasks. I started for many years, for almost 20 years, working on how the cortical circuits behave, what is the dynamics of these circuits, and how plastic they are as we introduce novelty or the animals, are performing new tricks, new tasks, they are learning something new, or there's some sort of lesion in the brain or in the periphery. This was a very important background that allowed me afterwards to derive what I call principles of neuroensemble physiology. Different from the principles of single neuron physiology, this was the first time that a series of laws that could be observed in animals and later in human beings allowed us to understand better the dynamics of cortical circuits and how they operate in normal conditions, not when animals are anesthetized or in artificial laboratory conditions. From that discovery came the idea that John Chapin, my postdoctoral advisor, and I had in the late 90s to create what became known, the name that we gave it to this new field, brain-machine interfaces. Because John and I thought that since we were recording large numbers of neurons at that point, perhaps to demonstrate some of our theories on how these populations of neurons encode information, we could link the brains of these animals to devices, to robotic devices, to computer graphics like virtual bodies or virtual tools, or even computers as a whole. And it was from this discovery and this invention of brain-machine interfaces that a lot of the things that I'm going to show you today came about, including a very comprehensive theory on how the human brain may work. That is very different from the theories that you guys see on your textbooks. So this is how a brain-machine interface looks like. This is the first engineering diagram that we published in 2000, showing that for a brain-machine interface or brain-computer interface to operate, you have to have a way to record brain activity, being intracranial with implants, or as you're going to see in a moment, in a non-invasive way with EEG or other techniques. And then you can record this raw electrical activity and process this in real time using very fast computers to extract from the raw electrical signals that populations of neurons produce, particularly in the motor cortex, extract from that activity the motor commands that the brain is generating to generate motion of the body. So we discovered that we could do that very quickly, even with old computers in the late 90s. We could do that in less than a third of a second. So we could actually record the brain activity, use mathematical models to extract the motor information that the brain is generating, and make robots move even faster than the body of the animal was about to move using the same command. So this is what is the main component, the afferent component of the brain-machine interface. But a very important second component is the feedback, visual, tactile, or even auditory feedback that we provide from these robots back to the brain, so that the brain knows how well it can control a device, either a mechanical device or a virtual device, just by thinking about the movements without engaging the body of the subject in this task. This was the first major accomplishment that we were able to demonstrate in animals. And this is just to show you the different generations of electrodes that were generated in my lab over the last 25 years to produce these recordings in animals, in rats and monkeys. These are very sophisticated, custom design for each area of the brain that we want to implant. And when you do that, as you can see here, this is on the left top, you can see a bird's eye view of one of these implants in an adult rhesus monkey that resulted in almost 2,000 neurons being recorded simultaneously. So this was, and to this day, is still the largest sample of individual neurons recorded with brain implants using this technology. So another great advantage that we discover on our method of making these implants to work in monkeys is that they could last for a long time. They could last for about 8 years. So if you look at this red line, what you see on the bottom in the x-axis is time in years. On the y-axis is the number of neurons on average that you could record in each day that we use this animal. And these 4 panels around the picture show you the quality of the single action potentials that we are getting from each neuron. And as you can follow, if you follow the red line, we were able to record this monkey for a long time. This is a paper that was published in 2014. So we're recording this animal for about 5 years at that time, but we were able to maintain these recordings until 2018 in the same animal. So there was a degree of longevity that nobody had seen before that we could achieve. So this is how it looked like when you did a Wi-Fi interface, a wireless brain-machine interface in rhesus monkeys. So in the right side, you see the cover of Nature Methods that this paper got, showing that the animals now were free. They didn't have any cables coming out of their heads. They could move around, and we could record the brain activity from these animals at a distance. As you're going to see, this was a major advantage for all of us. So, but let me show you a little bit of how a brain-machine interface evolved. This is the first brain-machine interface that worked in a rhesus monkey that was trained basically to use a joystick to control a computer cursor on a screen in front of the animal to play a video game. And that was very, very interesting because the animal played the video game as basically a child does it. One of our kids play it. And it was very interesting in winning the game. So in the beginning, the animal was using the joystick to move the cursor, and every time it crossed the target, the animal would get a drop of orange juice. And as the animal got better and better, we were recording, as you can see, the activity of the motor cortex of this animal while we performed the task. And we soon learned that we could basically, from 100 neurons only, extract the motor activity that was responsible for this joystick to be controlled by the animal. So a few minutes later, or a few days later, we decided to turn on this joystick and now transfer to the robotic arm that you see on the lower right of this figure, the control of the computer cursor that the animal had to move to play the game and win the juice. Well, that's how a brain-machine interface was put to work. We removed the joystick, so now the animal could use its own hand to play the game, but he still wanted to play the game to drink the juice. And we turned on the brain-machine interface. And when we did that, the animal needed now just to think. And he was able to control the hand of the robot to move the computer cursor, get to the target, and drink the juice. So let me show you how that happened. I don't even know what is going on again. But maybe, can you see this new slide?
Yeah, we can see.
Yeah, I can see.
Great. Yeah, there's a little movie here. Let's see if it works. Oops. What you're seeing here, I'm sorry, you went too far ahead. This is the movie, first movie. Here is the monkey playing with the joystick, the game. Okay, so you can basically see that it's very accurate. He can move this cursor very well, and he can basically cross the target and get the juice. And he's actually trying to guess before the target appears, this white ball appears, the monkey is trying to guess where to go, because if he guesses right, he will drink the juice faster. But while the monkey is doing that, we are recording the brain activity from this animal, and we are sending to computer screen, to a bunch of computers, I'm sorry, to extract the information that we need to make this brain-machine interface operate. So this is what is happening here. Look, the joystick is gone. The animal cannot play the game with the hand anymore. The joystick is removed. Now, the brain-machine interface is on. So every movement of the cursor that you see here is going to be produced by the brain activity of this animal going to a robotic arm that is next door, and now controlling this cursor to play the game for the animal. So the animal doesn't need to move its own body. So let me show you how this happened. Because this was the first time that a monkey liberated its brain from the limits of its own body to interact in the world just by thinking. So the animal is just thinking about moving an arm, but is not moving its own arm. It's controlling a robotic arm with its mind to be able to do this task. So this was 2003, about 21 years ago. Well, these monkeys got so much better with training that they basically reached a performance in which they could do this task like you're seeing now, as well as if they're using their own arms to move the joystick. So they didn't need to use the joystick anymore. And in fact, what we learned from that was a very different picture than what you see in your neuroscience textbooks in medical school. These graphs show number of neurons in the x-axis and how much on the y-axis each cortical area is contributing to that control that you just saw of the robotic arm. Each color represents a different cortical area. The navy blue on top is the primary motor cortex, the main area that is controlling the monkey's arms. Then you have in red the somatosensory cortex that is also involved in degrees of control, but is processing the tactile information that comes from the hands to the brain. As you can see here, all these 5 areas that I'm showing to you here have some degree of information about producing a motor program to move the arm. This was a very big news at that time, because people thought that only the motor cortex was involved in this. But look, the green area that you see here is back here in the posterior parietal cortex, which is connected to the motor cortex, but nobody thought at that time that it would be so much information available in this part of the brain to control the arms of the animals. So as you can see, depending on the parameter that you want to reproduce on our brain-machine interface, on the left side is position in space, on the right side is gripping force. All these 5 areas are giving information to our brain-machine interface. It's not only the motor cortex. So this is, as you're going to see in a moment, have profound clinical implications when we are talking about patients that suffer a lesion to the spinal cord or a lesion to the brain due to a stroke, for instance. So let me show you now a different brain-machine interface that not only allows the animal to use its brain activity to control a virtual arm and place this arm on virtual objects that are on a computer screen, but every time that the monkey places this virtual arm on top of one of these virtual objects, a different electrical current is sent back to the brain directly to inform the animal what is the microtexture of these objects that is touching on the screen. The monkey cannot see this texture on the screen because visually these objects look identical. But if the animal places the virtual hand on top of these objects, there is going to be an electrical stimulation that is proportional to the surface, to the microsurface of these objects. And the animal has to choose just one that has a particular degree of roughness that we told the monkey indirectly what to do. See, this is called a brain-machine-brain interface because the body was completely removed from any interaction with the world, both to touch the world or to feel the world. So what this animal is going to do then is try to do a tactile discrimination task without involvement of its fingertips or its hands. It's going to do everything just using a brain-machine-brain interface. And that's what you're going to see in this little movie. If I can get it. Oh, okay, here. Look at this. This is the virtual arm controlled by the animal, touching different objects that look identical. But I put some labels for you to see which one is the correct one that the animal has to touch. And when you hear this ping, it's because the animal got it right. He selected the correct object by basically pushing the center of this object when he finds the correct texture with the virtual hand. So the animal is doing that all with the brain. It's not touching anything with its own hands. It's basically doing that with this avatar of its hands, okay? Interesting enough, about 2 weeks later, this animal could do this identification of objects as well as if it was using its own hand. It reached the same level of performance just by using the brain to control an avatar arm and receiving the feedback of that avatar arm directly into its head, okay? But things got much more sophisticated. A few years ago, we got monkeys to learn to use a Wi-Fi brain-machine interface to drive an electrical car in our lab, to drive a self-driven car to collect some grapes that we put it in different locations of our one room in our lab. So these animals now were not just controlling a robotic arm or controlling an avatar, they were trying to generate commands to drive a car, an electrical wheelchair, if you will, that no monkey has ever seen in their lives. This was a totally new object that the monkeys had to learn de novo from nothing to control. It had nothing to do with their limbs. There was no biological relevance to a cart like that. And yet, these animals learned to do that. And as you can see here, first from a camera on the ceiling of our room, the white circles that you see are the departuring places where we place the monkey to start driving towards the red target, the location where the grapes are put in a pod so the animal can collect and eat the grapes. But you're going to see that the animals learn to drive no matter where we put them. They learn to generate a trajectory to take them to the grapes and eat the grapes. So that's from the top of the room, you can see the monkey driving towards the pod and collecting grapes to eat. Now we put the monkey in a new location and the animal just imagined a new trajectory, and that trajectory was sent to the engines of this the motors of this little cart, and the cart is driven towards the target. So look at the front now. You see that the animal only starts moving when it's getting close to collect the grapes because it has to use its own hand to eat. But it doesn't need to do any movement to drive the cart towards that pod. So at that moment, we realized, oh, this is just to show you that in the primary motor cortex of these animals, we found cells, individual neurons that code the external space in which the animal is navigating. You may have heard about these cells. They're called play cells, and they're located in the hippocampus. That's the classical discovery in the 70s. That was worth a Nobel Prize a few years ago in 2015. But here is the motor cortex, right here in the frontal lobe. It's not the hippocampus. And we clearly can find cells. Look at this. The yellow color means the cell is firing a lot. And the cell is firing in a particular location of the room as the animal is navigating to go to the pod. On the top is one neuron. On the bottom is a second neuron. And you see that they have different receptor fields for space. It's the same thing that you see in the hippocampus. And that was, as I said, the Nobel Prize of a good friend of mine, Dr. John O'Keefe from University College of London, that got a Nobel Prize for the discovery of these play cells in the hippocampus. But here we have them in the motor cortex, with nobody had seen it before. Well, all these results that I just showed to you clearly show to us that brain-machine interfaces could be used not only in experiments, but they could be used to help patients that have suffered devastated lesions of the brain, like as you see on the left, a cut of the spinal cord that makes these patients completely paraplegic or paralyzed below the level of the lesion. John Chapin and I, in 2002, on an article in Scientific American, proposed that if we use in the future a brain-machine interface like what we just described to you, we would be able to make these patients recover mobility by basically connecting their brains to a new body, not their paralyzed bodies initially, but a robotic device called an exoskeleton that these patients could wear, and under the control of their own brain, they could walk again, despite the fact that they had a lesion in the spinal cord, because we knew that the brain of these patients continue to produce the motor program that they want to send to the spinal cord to walk, but they couldn't anymore because there was a lesion in the nerves that take this information to the lower parts of the body. So that's when I decided, a few years afterwards, 10 years after that paper was published, I decided to create a project called the Walk Again Project. And I did that because around that time, Brazil was awarded the World Cup, the FIFA Football World Cup that was going to be held on June 12, 2014, the opening ceremony of the World Cup. And when I heard about that, I looked at the calendar. That was about 2 years before the opening ceremony. And I called the Brazilian president at the time and decided to ask whether Brazil would allow us to do a real experiment in front of 1.2 billion people that watched the ceremony by having a paraplegic young Brazilian to deliver the opening kick of the World Cup using an exoskeleton controlled by a brain-machine interface. But at this time, the brain-machine interface would be totally different. This is our lab that we built for the World Cup here in Brazil, in São Paulo. These are some of our patients and our original team at the time. On the left, you see the beautiful lab that we built here to create this technology and to basically train our patients for about a year before the World Cup to do that. A year and a half, actually. But this time, when I talked to our patients, they said, look, we are paraplegic patients, and we selected patients that had very definitive lesions of the spinal cord for many years. up to 13 years since the original lesion. And these patients could not move below the lesion. They could not feel their bodies below the lesion. They could not control the bladder or sphincters below the level of the lesion. But when I went to talk to them for the first time in 2013, they said, look, we don't want to have any surgical implants. We don't want to have a surgery. We want to do that without a surgery. And I realized, and that's the very important transition in my career, because I realized that despite the fact that I had invented these implants, and I had done a lot of studies in animals with brain implants, when it came to patients, neither the patients wanted to have that surgical procedure because of the risks of infection, rejection, bleeding, all sorts of problems that can happen in a neurosurgical procedure. But I realized that we could do what we wanted to do with no implant, that we could use a very traditional technique called electroencephalography that all of you may have heard already, just like a swimmer's cap, apply to the top of the surface of the head of these patients, place the electrodes on the scalp surface, and then extract the signals that we needed to control an exoskeleton for lower limbs, a robotic exoskeleton that we decided to build here in Brazil. And this is what you see on the right, is the entire training procedure that these patients went about, because we combined not only brain-machine interfaces and robotics, but we also use an immersive virtual reality environment to train these patients to regain the ability to control their legs or to walk again using a non-invasive brain-machine interface. So this is the first exoskeleton that we built. It's like a Ferrari of all exoskeletons because it's over-engineered to protect our patients the maximum you can, because you can imagine this demonstration had to be done in an open football field. Lots of electromagnetic noise, 65,000 crazy people in the stadium. And I have to tell you, you won't believe in this, but it's the absolute truth. We wanted to train these patients to, not be surprised by the noise that a football stadium produces when you're inside the stadium. And also the flashes of cameras, cell phones. So in our virtual reality, we put a lot of lights flashing while the patient was controlling an avatar himself. That was a perfect model of the exoskeleton, but we needed noise. We needed noise of fans to distract our patients. And we did a worldwide survey of who could be the noisiest football fans in the world. And I'm from Brazil. I thought the fans would be Brazilians, but you won't believe it, but it's the truth. The Turkey fans won. So we put our patients to train under the sound of fan clubs in Turkish stadiums during the derby, between the big teams in Istanbul. That was the sound that these patients heard to train for the opening ceremony of the World Cup. The sound produced in a Galatasaray and Fenerbahce match in Istanbul, okay? So you can say that Turkey was present to our demonstration 10 years ago. So this is one of the most important components of the exoskeleton. This is what my good friend Gordon Schenk, who invented this in Munich, in Germany, calls artificial skin. These are sensors that you place on the foot, on the plantar surface of the foot. So every time the exoskeleton touches the ground, a pressure wave is generated, and we can deliver using cell phone vibrating elements to the forearm of the patient. So the patient can feel that the exo is touching ground. So not only he can see the exo moving, but he can have a tactile sensation of walking. And this was very important to retrain the brain to acquire the feeling again that he had a full body to walk. So this is the first time one of our patients actually walked using this brain control exoskeleton. You saw a blue light on top there. This was because the patient's brain was producing the commands to control the movements of the exoskeleton legs. And you can see that he's looking straight ahead because there's a big mirror in front of him, so he can see himself walking. For the first time in many years that he was paraplegic. This patient had a lesion at mid-thoracic level, so he could not walk or move any muscle below this level and could not feel its body or his body below this level. And now, after all these years, he was walking for the first time using a brain-machine interface to control this exoskeleton. I don't have the images of the opening ceremony because FIFA never gave them to me, so I cannot show you the actual images, but this is just a few seconds before we went to the field on the ramp of the stadium here in São Paulo, and this is Juliano Pinto, the patient who delivered the opening kickoff of the World Cup. And what I'm going to show you now is Juliano asked me to give one more try before going to the middle of the stadium because he could hear the noise of the Brazilian fans there. He was a bit nervous. So what you're going to see is my student, Suleiman Shakur, who today is a professor at the APFL in Lausanne in Switzerland, placing the ball, and Juliano is going to use the brain-machine interface to make the exo kick the ball. So Suleiman is putting the ball in there, and in a second, you're going to see Juliano kicking the ball. And that was done by his brain producing the command, and the interface, the brain-machine interface, reading the intention of Juliano to kick the ball and send the ball rolling, as you saw. And when he did that on the soccer field, we had basically turned on a pressure sensor right on the top of his toes, on the foot, for the first time. And Juliano, when he kicked the ball, he received a message in his arm of the contact with the football. But his brain interpreted that message as his foot, his real biological foot, kicking a football again, something that he had not done in more than a decade because he had a very high lesion on the spinal cord. But this day, he felt the ball again, thanks to this tactile feedback. Well, after the World Cup, we brought these patients back to the lab. But what we didn't know is that something very unusual and unpredictable had happened. When we went to examine Juliano 1 month after the kick in the World Cup, Juliano was originally classified and remained for 10 years as a T4 paraplegic, meaning his lesion was a thoracic level 4. After 1 year of training, and after 1 month after the World Cup kick, we redid the neurological examination in Juliano, and to our shock, he was reclassified as a T11. Out of nothing, he had gained 7 dermatomes below the original level of lesion, not only of sensitivity, but also of motor control, partial motor control. He had experienced a partial neurological recovery that had never been reported in the literature, in the medical literature of spinal cord injury. So we are very surprised by that, very shocked, decided to stay quiet and repeat, this neurological examination in all 8 patients and follow these patients for 20 more months. And to our shock, as we follow these patients, their recovery just improved. So after 28 months of training, the 7 patients that remain in the protocol, we could see an average recovery of 10 dermatomes below the original level of the lesion. So these patients were being reclassified from complete total paraplegics, what we call in the American classification Asia A, to partial paraplegics, Asia C. They had been updated to a clinical state that nobody had been able to demonstrate in chronic paraplegic patients before. And let me show you how that looked like. This is after 13 months of training in one of the ladies of the protocol that had 14 years of lesion at the time. She had stayed 14 years as a complete paraplegic. One day, we suspended her and asked her to try to do whatever she could. And all of a sudden, she started moving her leg, her right leg, something she had not done in 14 years. She did that for a full hour. And we were, again, stunned because we didn't expect some neurological recovery like that. Well, we waited one more year and tested these and other patients again. And what we saw in the same patient a year later was even more shocking. When we asked her again in the same task, but now we are recording EMGs all over her legs, to move as best as she could, she did this. She first moved the right leg as she had done the year before. But suddenly she was now moving the left leg. And as you can see, when we pull her back, she started doing the same sequence of right and left leg movements that we could not see a year before. And she could not have done it for 14 years. So we had a clear demonstration of a partial neurological recovery in a patient that was originally classified as a complete paraplegic. Well, as time goes by or went by, every one of the 7 patients that remain in our protocol were able to make that transition. It took different times for different patients because the level of the lesion is different. But at different moments in time, every one of the 7 patients moved from complete paraplegia to partial paraplegia. One of the patients left the protocol at 12 months, so we didn't have a chance to follow him up, but all the other 7 patients did the same thing. The recovery in some of these patients was so spectacular that a few years later, 1 or 2 years later, some of them didn't even need the exo anymore to walk. They could walk by themselves just using a little walker to give balance, to remain in balance. And as you can see, this patient is wearing the swimming cap that I mentioned to you before, because we are using a brain-machine interface to record his brain activity and to use that to stimulate a few key muscles on his legs, just to give some help for him to walk by himself. But most of the movement is being produced by his own legs, by his own voluntary motor control of his own muscles, because he had a very significant partial neurological recovery. So he didn't need to have an exo to walk anymore. So this was 2019. Of course, we had a lot of issues with the pandemic, but before the pandemic, for the last 20 years, I had built this facility here in Brazil with the support of the Brazilian government, which is now known as the Campus of the Brain of Brazil, located in the northeast part of the country. And in this place, we have a neuroscience institute that is primarily focused on brain-machine interface research. And in that place, we have created a very modern center for neurorehabilitation that we are using to expand our protocols and our therapies. And I don't know if you have seen it. I didn't put in this presentation, I forgot, but just a week ago, we announced a partnership with the largest private hospital in Italy, in the city of Milan, to build the first European neurotech hub, basically to replicate this center that we have in Brazil, in Europe, to create the largest center for neurorehabilitation in the European continent, to basically do exactly what you saw here, but to many other types of brain disorders, not only spinal cord injury, but stroke, chronic epilepsy, multiple sclerosis, and a lot of new disorders that may benefit from brain-machine interfaces. In fact, this is our center here in Brazil still, but 8 years after we published the original paper that I show you in 2016 with these eggs on the left, we have now manufactured the third generation of this device, which is much simpler, much cheaper, and much easier to use, and is controlled just by a cell phone. You don't need all the electronics that we had in the backpack of the original exoskeleton to produce the effects that I showed to you. We can basically use just a cell phone. This is one of our technicians, one of the computer scientists, I'm sorry, in the United States who participated in this project. And he's basically using our brain, a non-invasive brain-machine interface and a cell phone to walk with this exoskeleton. He's not a paraplegic patient, he's a healthy human being, but he was just doing a benchmark testing. He's relaxing his body and he's letting the exoskeleton carry himself. But as you can see, he has a much simpler electroencephalography interface on his head, and he's using a virtual reality training program on the iPhone to basically read his brain activity and to control the exoskeleton, which is much more affordable, much cheaper, and can be produced in large numbers and can be used by a large variety of patients right now. So this is how we evolved from basic science all the way to this level. But I wanted to show you one final example because as I created my new institute here in Brazil a couple of years ago, I created a new project. In addition to the Walk Again project, we launched with our partners now in Europe and with partners around the world. Who knows, one of these days we may partner with you guys in Turkey. But we are basically launching the Treat 1 Billion project. Why Treat 1 Billion? Because the World Health Organization has estimated that more than 1 billion people around the world have some sort of brain disorder. And most of these disorders cannot be treated effectively with medication, with other therapies that we have. Well, I truly believe that non-invasive brain-machine interfaces may be the basis of a new generation of therapies that can be used to treat 1 billion patients around the world. And in fact, with our partners around the world, we are going to implement this generic, non-invasive brain-machine interface to treat a variety of these disorders. And I just want to show you, to finish, one example, also from basic science all the way to clinical practice, that is going to be one of the main targets of this new project in Milan, Italy, and here in Brazil. In 2009, I published this paper with my students that was in the cover of Science, because we had shown that when we look at transgenic mice, they were at that time thought to be very good models of Parkinson's disease, in particularly a condition called freezing of gait that some Parkinsonian patients suffer, in which they froze. They completely froze. So they cannot move. I mean, they are basically freezing as they try to move. Well, when we look at the brain of these animals, what we realized was that the activity of these neurons in the motor system, in the basal ganglia, in the motor cortex, look like a seizure, epileptic seizure, in which most of the neurons synchronize in this frequency around 10 to 20 hertz, which is known as beta frequency. When I look at that, since I have worked with another student in epilepsy before, I said, you know what? Let's stimulate the spinal cord, the dorsal part of the spinal cord, with about 150 hertz to see if we can disrupt this seizure and get these animals to feel better, to move, because they're frozen right now. My students didn't believe me in the beginning. They thought that I had lost my marbles here, but they went ahead and did the experiment. And long and behold, when they stimulate the dorsal column of the spinal cord, right on the surface of the spinal cord, the seizure went away, as you can see, in the blue region here, and the animals move. Let me show you how. So you see here a frozen rat that is totally Parkinsonian. He has lost 99% of dopamine in its brain. But he wants to go to the lower right corner of the cage because he knows that there is sugar water there for him to drink. But he cannot move. Well, in 5 seconds, we are going to start stimulating the spinal cord as I told you we did, right now. And look what happened. Instantaneously, the animal can now move, even though it has no dopamine left in the brain. That is happening because we kind of reset the brain of the animal. Like when you have a heart attack, ventricular fibrillation, and you give a mega shock to desynchronize the heart muscle fibers, we here desynchronize the animal's brain, and he could move freely. Well, some friends here in Brazil saw this and decided to test this idea in humans. They implanted 4 patients with a spinal cord stimulator. This is an epidural stimulator, commercially available. These were patients that suffer from the same condition, freezing of gait. And they basically did exactly what we did with our animals. On the left side, you're going to see these patients just with DBS and no stimulation of the spinal cord. On the right side, you're going to see the same patient the same day when we basically stimulate their spinal cord. Oops. And you can see is this. On the left, this woman could barely stand up. She was frozen. She couldn't move. On the right, we start stimulating her spinal cord with the same stimulation that we did in the rats that you saw before. And look at the difference. She can move. She now can walk. What, took her 20 meters to walk, took her about 5.5 minutes, she can now do in 57 seconds. 5 times, more than 5 times faster, without the help of the nurse, because we have disrupted the seizure activity that she had in her brain as a result of Parkinson's disease. You're going to see the next patient. I told him that he was ready to play football now, because you're going to see the reaction of his brain when we did the same thing with him. On the left, again, no stimulation of the spinal cord. On the right, stimulation of the spinal cord. Look at the difference. It's pretty impressive. You're going to see in a moment the same 20-meter task on this guy, and you're going to see the same effect. He can now walk without help. He can make turns. As long as the spinal cord is stimulated once every 3 days for an hour, he can do that. And more recently, we have realized that we don't even need to implant the spinal cord. We can do from outside using a transcranial magnetic stimulation on the surface of the skin of the cervicothoracic location. That's the region of the spinal cord that gives you this effect. So even though the spinal cord is not involved at all with the physiopathology of Parkinson's disease, this type of neuroprosthetic device may become, a very efficient treatment, even for the cardinal symptoms of Parkinson's disease. So right now, we are connecting this technology with a brain-machine interface, the generic brain-machine interface that you saw a few slides before. This one here. And we are going to allow the patient's brain activity collected by a non-invasive EEG method to trigger the stimulation of the spinal cord when the patient feels that is about to become frozen. So he's going to control the flow of stimulation of the spinal cord to allow the Parkinsonian symptoms to go away. So this is a brief summary, just 50 minutes to show you how we went all the way from a totally basic science program to study how populations of neurons encode information, and found out yourselves creating new therapies that may serve a huge number of people around the world that suffer from brain disorders that when I went to medical school in the early 80s, my neurology professors told me we would never find cures or therapies for these patients. Well, I'm celebrating 40 years of my graduation from medical school this year. And of course, my professors are not here for me to tell them, but I have been able to live through these 40 years and see the moment in which we may be able to treat patients that we thought we would never treat once in the past. Thank you.
Thank you so much for your presentation, Professor.
My pleasure. Yeah, I'm just going to cover a little bit of my video just to stop the sun coming in my face here because the Brazilian sun has betrayed me.
Okay. Now we will begin the Q&A session. So please feel free to type your questions or contributions to the chat box. And we'll strive to address as many of them as possible during our Q&A session.
Well, first, I would like to ask something. First, thank you for your great presentation, Professor. And I'm really amazed about the contributions you made to the world of neuroprosthetics. And also a bit amazed by our country's sports teams, fun clubs. So I would like to ask, does the efficacy of implants change by doing it with surgery by directly to the brain and without a surgery and doing it with EEG? So how does this affect the efficacy of the exoskeleton?
You need to rephrase it to me because I didn't quite get it. There is a question here. I don't know if it's exactly yours, but someone is asking me, how could we do this without a Faraday cage? Yes, we could. We could do recordings. We did it in a football field, open, because we had a little secret that we developed that is shielding our system of EEG system. So yes, you can do this without a Faraday cage. But please, could you rephrase your question so I can get a better sense of it?
So I was wondering the effectiveness of the implant changes if it's with a surgery or without a surgery and doing it with EEG.
Let me tell you first. When you do a EEG, it's not an implant.
Okay? Yeah, it's not an implant. So I'm asking, does effectiveness changes?
Well, of course, if you're dealing with animals, the resolution and the amount of information that you get inside the brain is much higher than EEG. Mm-hmm. However, implants, as I said before the talk, they have risks. They can infect, they can stop working, they can lead to bleeding when the surgical implant is being made, and they can produce rejection. They can produce tissue rejection, inflammation of the tissue around. And although in monkeys, we have been able to keep these recordings going for 9 years, because we use a particular type of metal and a particular type of very flexible metal In humans, nobody has been able to reach that long or that longevity. In fact, some of the rigid electrodes, the Utah probe, don't last more than a few months or max a year or two, okay? And that is not enough, because once that stops working, you cannot go and remove it, okay? That's a big problem, because if you try to remove it, normal tissue will come, okay? So the big advantage is, first of all, safety, which as a physician, you need to put in a very high level because you're dealing with human beings. And as you know, because you're all from Turkey, very close to Greece, the Hippocrates Code says that the first thing you should not do to a patient is to increase their problems, right? You should improve their quality of life. So, number two is efficient, meaning, as you saw, we can make people walk, we can treat their problems, we can train them, and we can share the control of these devices between the brain of the patient and the robotics. It's called shared control. And that's exactly what we invented 10 years ago here in Brazil, because the control was the voluntary decision, I want to walk, was given by the patient. But the robot took that decision and did all the micromanaging of the motors that were necessary for the walk to occur, okay? And there's a third component that is very important. A surgery like this, a neurosurgery to implant these electrodes in the United States, is going to cost between $100,000 to $200,000.
Wow.
That's the reason some companies want to push these implants, because they want to make money. This EEG system that you're talking about is a couple $100, not thousands, a couple hundred. It's cheap, it's inexpensive, so we can scale up to 1 billion people. I implant, nobody ever will be able to implant millions of people. It's not going to happen, okay?
It's really hard.
Yeah, it's a little difficult. You need hospitals, you need surgeons, you need money in large quantity, and you need to have people that can afford to pay for it. We don't have that in Brazil. I'm sure you don't have that in Turkey. You don't have that in China. You don't have that in Europe, okay? And not even in the United States. So I'm not doing that as a distraction because I'm a billionaire who have nothing better to do with his life. I'm doing that because I invested 40 years of my life into this. And I want to reach a huge number of people. That's a big difference. You know, you asked me in the beginning, what is the difference between what we do and what Neuralink, a company that my former students created? The difference is I'm not a student. I'm the professor who created, and I want to take this to a huge number of people, not make money out of it.
Yeah, I can understand it clearly right now.
Thank you.
So we can proceed to the questions in the text box, Sena.
Okay, I can read them if you want, Professor.
So should I answer the questions that I'm reading in the chat, or what should I do?
Yeah, if you can see them, you can answer them, of course.
Yeah, I can see it. So I can go very quickly to them. You know, very good questions, by the way. Yes, the stimulation that I applied to the mice and my colleagues did in humans, they need to be repeated at regular intervals of about 72 hours. So it's like a massage. You do a back massage for an hour, and then you're good to go for 3 days. But then you need to do it again. But there's one important finding. If you do that continuously, every day or every 3 days, in animals at least, we saw that slows down the death of dopaminergic neurons that are still alive in the motor system of these animals. It's almost like you could slow down the progression of Parkinson's disease if you do that. That's the hypothesis that we are pursuing now. Another question. Yes, it is possible, and people have already done it in many places in the world, to use non-invasive brain-machine interfaces for rehabilitation of stroke victims. You don't need to put chips in the head to do that either. Mr. Musk is not going to be happy, but we can do that, and we are going to do that in Milan without implants. That's the two questions I can see here so far.
Yeah, we haven't got any other questions yet.
That's okay. There's one here. Which part of the spinal cord do we stimulate? Yes, it's a special location. That's a very good question. It's very critical. It's in the cervical thoracic transition, a little more to the cervical enlargement region. So C7 to T3, about that. It's not special neurons. It's because this is where you have the largest density of fibers that are going up to the brain. In the dorsal part of the spinal cord, the part that is tactile. Is that okay?
Yeah, thank you for your answers, Professor.
Oh, my pleasure.
We can proceed to end the meeting, Sana.
All right. Are we going to take a picture?
Yeah, it would be great. If you can open your cameras, we will take a picture to put on our Instagram. So let's wait a bit. All right, then I'm taking the photo. 3, 2, 1. Yeah, I took it.
Great.
Okay, thank you so much, Professor, for this presentation. That was so incredible.
Thank you. It was a great pleasure talking to all of you. I wish you all the best and get some sleep because it's late already.
Thank you,
Yeah,
Professor.
thank you so much.
Thank you.
Thank
Bye
you.
bye.
Thank you so much.
Goodbye, Professor.
Goodbye, Professor.
Bye bye.